And as our program states, we focus on Crohn's disease, and we are interested in actually launching and starting the Gut Cell Atlas. Some of the results were presented today, and as you saw, we are also interested in the Gut Cell Atlas roadmap that we started brainstorming about a couple of months back, and hopefully with the help of many of you here and others in the field, we'll be able to design and develop and hopefully implement a roadmap with clear goals and outcomes set beforehand, as Dana mentioned earlier. And we are excited to be here today to discuss about some of the questions that we circulated, and I think you all have on your computers. Sorry, we don't have printouts. And without further ado, I think what we could do is just to start a quick intro to understand why you are interested in the gut, and eventually if you are working on the gut, and hopefully this will be an interactive discussion and brainstorming session. Is there a microphone?
I don't know if we got a. People might just have to shout. People might just have to shout because we weren't given a roving mic. So just yell a bit. I think it's not such a big room. I don't know if you want to introduce yourself.
Okay. So my name is Alexander Denadai Souza. I'm a senior postdoc at K11, and I'm working in the lab of Professor Guy Buxtens. Target, so we are primarily interested in gastrointestinal disorders like IBS. And we recently, I'm happy to announce that we got a grant from Hemslake. We're having the kickoff starting meeting late this month. So in this project, we are going to characterize macrophages, especially resident macrophages that interact with enteric neurons, because we recently published in Cell a paper demonstrating that these resident macrophages somehow nourish these neurons. And once we deplete these macrophages, we have loss of this neuronal enteric population. So there are many consequences. So this is a project that I'm not really directly implicated. The second part of the grant we got is to study enterochromaffin cells in IBS, but also in Crohn's disease patients in remission, but that still have IBS-like symptoms. So we performed a proof of concept study. We know that enterochromaffin cells are highly modified transcriptionally in this condition. So now we're going to re-challenge this hypothesis of ours that enterochromaffin cells are the main cells implicated at least in pain in these conditions. So we're going to do 10x of the whole epithelium of health volunteers, Crohn's disease patients with or without IBS-like symptoms.
Thanks.
Great. Hi everyone, I'm Kylie Leotono. I'm at the Broad Institute in the Data Sciences platform. I work with a team that makes the pipelines for the... thank you. Hello? Great. I work with a team that makes the pipelines for the Humans Atlas data coordination platform, and I'm also your scribe today.
Hello, I'm Irene Papatheodorou. I work at the European Bioinformatics Institute, and my team deals with analysis of single-cell RNA-seq and development of Expression Atlas, a resource for transcriptomics. And here we're interested because we collaborate with a group at Edinburgh led by Mark Arens on the Human Gut Cell Atlas and Crohn's Disease Atlas.
Hi, my name's John Manning. I'm a bioinformatician in Irene's group, actually, so ditto.
Hi, so I'm Azucena Salas. I'm an investigator here in Barcelona, and my group works with inflammatory bowel disease patients, and so we just recently, literally the last year or a little bit longer, have been actually using single-cell RNA-seq to look at the biopsies from these patients, so this is an area that I'm very, very interested in. We're learning as we go.
I'm Sara Jimenez. I'm a PhD student at the IGBMC Institute in Strasbourg, in France. And we are working, we are using single-cell techniques such as RNA-seq and ATAC-seq to understand the differentiation process in enteroendocrine cells in the small intestine.
My name is Chen Hui Yu. I'm postdoc of Greg Camp and Barbara Troutland. And working together with Jason Spence in Michigan University, we are using the single-cell technology to investigate the development trajectory of the organoid derived from iPS cell. And also, we would like to compare the organoid with the fetal intestine and other fetal organ to really see the intestine-specific features and how these features established during the organoid development.
Hi, I'm Louisa Moore. I'm a pathologist from Cambridge, and in my clinical practice, I'm interested in gynecological disorders, not GI. But in my research practice, I'm based at the Wellcome Trust Sanger Institute, working with Mike Stratton and Peter Campbell, and we're interested in using not single-cell approaches, but low-input laser capture microscopy-based approaches to infer somatic mutation and clonal evolution in normal tissues. All normal tissues, including the gastrointestinal tissues as well.
I'm Henk Stoeneberg. I'm at the Princess Maxima Center for Children Oncology in Utrecht recently. We're working on celiac, we're working on colorectal cancer, and with the gentleman two further down on the organoids in the human project of the Human Cell Atlas from the EU that has just been approved.
Okay, I'm Young Park from the professor of Samsung Medical Center in Korea. And we are currently working on the colon cancer single-cell RNA-seq project in Korea. And so we summarize about 30 cases in the normal tumor pairs and then starting metastasis pairs for the colon cancer too. And also expanded to other esophageal and also other stomach, and then maybe it's aligning that GI, the cell, single cell project. And also we work with the other Belgian group, and then to compare any ethnic difference in the combining with the microbiome data together.
Hi, my name is Tae-hee Hwang, and a professor at Seoul National University in Korea. And recently, I'm initiating the single-cell analysis of the using gut tissues, and so I came to this breakout session to hear about what are the important issues in this area. That's it.
Hi, I'm sorry I came late, and now I'm realizing that we must be introducing ourselves, so I picked the wrong seat to sit. So my name is Jeff Moffitt, and I'm an assistant professor at Harvard Medical School and Boston Children's Hospital. My lab is using MERFISH and other spatial transcriptomic and proteomic methods, and we are very interested in questions associated with the microbiome host interface.
My name is Farzin Purfarzad. I'm working in Hubrecht Organoid Technology. Hubrecht Organoid Technology is a foundation that is a spin-off from Henk Stoeneberg Lab. And basically what we are doing, trying to use the organoid technology and expose it for industrial and big pharma drug development services. At the moment, we are involved in generating of human cell atlas from 100 colon organoids together with Henk Stoeneberg. And in other activities that we are doing, we are generating organoids from IBD patients and trying to develop a platform that we can use organoids as a screening model for drug development.
Hi, my name is Genta Nagai, and I'm from the University of Tokyo in Japan. And my research interest is liver and gastric cancer. And we have studied the single-cell analysis of liver cancer model using mouse model.
Hi, I'm Giovanni Iacono from Senac CRG here in Spain, and I'm the live stream moderator.
Hi, I'm Jerry. I'm a computational postdoc from the Broad Institute. I'm interested in to develop some algorithm to analyze mostly such as single-cell RNA sequence data for bunch correction, visualization, and also for lineage tracing. I am also interested in the gut because now I'm also working on to quantify the different cell types, cell states, in the small intestine and also in the human esophagus.
Hi, my name is Yoshiaki Tsunami. I'm from Germany, Halle an der Saale. So yesterday it happened something really bad. Maybe you watched the news. So we are working in the surgery department in the university hospital, and we are interested in mainly the pancreatic and the liver cancer. And then we are also running a biobank system, so liver, pancreas, and so colon cancer, and then also the gastric cancer. Yes, so this time, so there is no also not the pancreas or liver cell atlas, also not the major focus. I mean, we are doing also the colon cancer, but gut cell atlas is not the major focus. But I'm interested in as well. I'm happy to discuss with you.
Hi, I'm Fiona Griffin. I work at the Chan Zuckerberg Initiative. I'm a project manager there for single cell biology, and I'm here because guts are cool. So.
I'm Sanja Vikovic. I work with Aviv at the Broad, and I mostly work on spatial organization in the gut. I work on gut development and aging, and I'm basically working with a lot of questions that Aviv and Orit presented today, and on top of that, I work on spatial host microbiome interactions similar to Jeff's interests.
Hi, I'm Moshe Biton. I used to be a postdoc at Aviv Regev Lab. Did a lot of work on the gut. I'm interested again in the gut because it's a very cool system. As they say, it's the central of our body. And of course, I'm very interested in epithelial immune interactions, trying to understand a little bit the normal gut, and then of course, different diseases such as inflammatory bowel disease and colorectal cancer. And I'm now in the Weizmann Institute.
Hey, I'm Mike Schnall-Levin, head up R&D at 10x Genomics, and I'm just here to learn what you guys are doing in more detail and hear what you guys want from us in the future. So thanks.
My name is Jose Ordovas Montañez. I've just wrapped up my postdoc in Alex Shalik's lab. And I'm starting my own group over at Boston Children's Hospital, where the main focus is going to be understanding general principles of how inflammation shapes 3 barrier tissues. And so I had to pick my favorite one, so I came to the gut, over the airway and the skin. But I might duck out early to see what's going on somewhere else.
Do you want to present or?
Okay, I'm Marta Melé. I'm a new group leader at the Barcelona Supercomputing Center. I've done a lot of work on transcriptomics, but I'm now diving in the single-cell transcriptomics, and so I'm here to just see how cool the gut is.
Great. But I thought I'd just, since I got the floor, I'd kick off very briefly, because it's not meant to be another talk, but just with something technical, which we've been, sorry. Oh.
Oh, so let's give him a mic.
So hi, my name is Joan Aruga. I am working as a software engineer at Red Hat in Czech Republic. As my second private project, I am working for gut microbiome analysis project.
Okay. I was going to show just a few technical slides, because just to kick the thing off, and there's obviously a list of questions, so we can get through these. These may or may not be relevant, but one of the projects that we've been doing is on celiac disease, which is kind of straightforward in some ways because it's pretty localized. And our approach has been, for this project, has been to biopsy patients who are undergoing regular surveillance anyway, and then digest the biopsies, make single-cell suspensions, and put them through the 10x sequencing platform with help from Sarah Teichmann's group at the Sanger. And basically, because it's an immunology lab, our focus has been primarily on the T-cell side and other leukocytes, but we're also going to apply the same approach to epithelial and stromal cells. So what we wanted to do to streamline the workflow, and these things were all addressed to some extent in the previous session, both the issue of how you handle controls and patients, whether you do them separately or together, and the issues about batching. We thought it would be a lot easier if we could freeze our samples, and then we would have some control over who we actually put forward for sequencing, given that we couldn't sequence everybody, and also we'd be able to match things a bit more effectively and batch the workflow. So this is a small experiment that we've done, which has sort of revealed to us that it actually works okay. So we basically, as you would imagine, took the same sample, froze it, or did things fresh, basically, so cutting out a lot of the analysis, you get quite good retention of the main populations that at least we were interested in. So it's important just to recognize that we're only looking at the CD45 positive cells here and not the other populations. So with the fresh and the frozen, that the populations are all pretty well represented. So we didn't lose anything in particular from what we could see. This is just reproducing data that has been already kind of generated using flow cytometry and to some extent qPCR. So it wasn't a big surprise, but it was kind of reassuring. The actual UMI counts and so forth are relatively well preserved between the fresh and the frozen samples. So that was also pretty reassuring. So that's a reasonably systematic look. And then basically, I guess what people would want to know is whether the cells cluster differently if you take them through the process fresh or frozen. And as far as we could tell, I mean, you can do these analyses lots of different ways, and this is only small numbers, but we didn't get a big deviation between, in this case, we're looking at CD8s, which is our favorite cell type. And they kind of correlated as reasonably well in terms of the things that they were expressing. And then I guess this is probably the most important slide. I think there are subtle differences between the fresh and the frozen cells, but it doesn't seem to interfere a lot. So in other words, it's the principal components. They don't lie entirely one on top of the other, but they don't deviate substantially and essentially create big deviations so that you can't assign the population. So we were reasonably happy that the cells that we assigned from the frozen biopsies are more or less the same as the ones that you get from the fresh. And I should say, the protocol is all up on the protocol's website. And oh yeah, I'm not sure. I didn't go back and check, but they're I mean, these are just basically the principal components from the whole data set. So if you take the principal components with the frozen data set and from the fresh data set, they're broadly the same. But if that makes any sense, there'd be people in the audience who know much more about the algorithms than I do. But fundamentally, that's PCA, a simple way of just overviewing these. So I think it's just given us some confidence that we can batch the things. But it's also incredibly simple, so that you don't have to separate the cells up front. You just freeze the thing as a biopsy, which surprised me a lot, but it just seemed to work. So you put it in some freezing mix, and then you can kind of organize your work around that. So since one of the questions was around fairly technical things, I just thought I'd show those slides, and then people can take the conversation forward as they want. So thanks. I don't know if anybody else is doing that, because I think, yeah, should we, I'll do the...
Thanks. I think we're basically running the same project as you do, but have started this already quite a while ago. So the data is on sorting 384, but the pictures are, and the results seem to be, as far as you showed them, quite the same. And at the 384 protocol, I think we see more batch effects than we would see on the 10x, which we're now routinely using. So basically have stopped doing the 384 because of the more stronger batch effects.
Thank you. So I assume that you took the biopsies, so you have the other compartments as well, the epithelial and stromal cells. Yes. What can you say about them?
Yeah, we haven't done that much. So I think this is definitely, there are ways to do it, because Alison Simmons, who's published the nice work on the epithelial cells, that for some of their analyses, they did use frozen biopsies to maintain those, to extract the epithelial cells. Our concern is that the protocols are slightly different. So when we take the cells out, we might have to devote one set for one and one for the other, but I think that's probably a price worth paying. I mean, the other way would be to try and aggregate everything together, but then it's a bit of a compromise for both sets. I think it's a good solution.
Actually, our standard protocol for the colon cancer project is based on the cryopreservation of all the samples. So we preserve about 200 cases in the dissociation in the freezer, because we cannot accommodate all the money for that kind of cases. But the problem be actually we check in not every month, but actually when we check in the 6 months later, the preservation, the cell population a little bit changes. So maybe it's gonna be to check that how old is that the samples. And the other thing, actually, when we checked in the PBMC, the cryopreservation protocol, the granular cell population is changed. So maybe it's gonna be to check that cell types, but depends on the tissue type. Actually, it's, sometimes it is that we miss some population in the cryopreservation.
I just wanted to add, actually, there are some groups that are doing full tissue or biopsy, at least in the gut, full biopsy, sort of cryopreserving them and then dissociating later on, which for some cells might work, but not for all cells. So has anyone tested that, and do you think that's an option? Because it's true that collecting samples, especially from a big organ like the gut, it's time sort of, you have to be time sensitive because it gets degraded. So how can you eventually process so many samples at the same time and eventually do all the facts analysis or whatever you want to do afterwards? So if anything.
That protocol was, that protocol was based on freezing an entire biopsy, which seems basically, I guess there's maybe an optimal size of biopsy that you might be able to achieve, but with just a regular clinical one, that works reasonably well. So, and that's only in the upper GI tract, but there may be more experience. And I agree with the comment about the timing. I think that's been shown elsewhere, that it works well for a bit, and then we might just have to use the samples.
Yeah, so what we're starting to do is to take nuclei instead of cryopreserved over longer time. Nuclei works perfectly fine, even on the cell types that are notoriously bad, like neutrophils. You get good data out. So I would suggest that nuclei-seq or nuclei attack-seq is perfectly suited in these cases.
I think just for practicality, what we'll be doing, I'm going to pass microphones on both sides and maybe just pass them along to, it's easier for both of us because we have to report back. So I think just to kickstart and maybe going back to Dana's presentation, any thoughts on what or how can we define the goals and why are we studying the guts and why to build the guts atlas? I would love to hear from from everyone just to start thinking in this direction and eventually defining outcomes. What are we looking for? So what are the questions that? So any thoughts?
So maybe from an outsider point of view, because I'm not in the atlas, but I'm looking more in the disease, I was surprised to hear, a lot of your concerns on the finding the rare cells, which I think maybe is one of the main goals of an atlas, from the point of view of somebody that just is interested in the disease and maybe not so much on a whole organ description. I'm also more interested in getting more depth within each cell, which I think is one of the limitations I'm finding, at least when we're doing, the 10x on the sample. So sometimes the questions are, from the point of view of our patient, is these cells that change, what is the entire signature of the cell? And I am finding limits on how many genes you get per cell. And so sometimes even like, there was a presentation on the covariance, I was trying to do analysis like to correlate genes, like if these type of T cells are expressing this transcription factor or whatever, what else is there, that type of analysis. I mean, a more practical, application of the data. So to me, maybe it's not so much the goal is to find the most rare cell as to know more about the cells that are changing in the patient and what is the whole signature of the genes in those cells. And
Can I make three maybe points that are relevant? The first is that it can be rare cells that are changing. I mean, we've seen that in ulcerative colitis in what is now a published study with M cells, which are not even colonic cells typically. Well, in the colon, they're very low frequency. In a bulk sample, how do you detect them? You might see some of their expression. In any case, my point is that cells can be relevant to the disease and rare. The assumption that the rare cells are irrelevant to a disease, I think, is not a very safe assumption in diseases. And also the term rare is a numerical term. So for one person, 1% might appear to be rare, another person might consider an 0.1% cell to be rare. So that's another consideration to take into account. There's a difference between rare cells that you know are there and you can go for them and those that are not. So that's kind of one item. The second was about asking on the content inside the cells. So one of the main two things about these lower depth assays, their goal is not to ascertain the level of each gene in each cell. Their goal is to give you a characterization of the cells as groups, as communities, as whatever. You can always bulk it back up. You will get those rarer transcripts, such as those for transcription factors, but you wouldn't be able to say about each cell and its variation. And then you go to what Dana described, these covariation patterns and so on, things that are more robust. So that's one path to get there. And the other is, which is also highlighted in the white paper, is that it is very useful to also have bulk samples that are analyzed, say, by deep RNA-seq, so that you can get a general idea of the transcriptome that is there. And then when you put both of them together, you can kind of apportion what is going on. So some of these are analytical solutions. They don't actually change the experiment that you do, but they change the way in which you look at the data. But I would be cautious to say, well, we already know the cells that matter, and they're abundant, because there could be plenty of cells that are not abundant. And if you look at a bulk sample, and you see a change in gene expression, you can't actually say whether that change is those cells changing their expression or the proportion of cells that is changing. That is by now, I think, just experimentally shown. It's no longer an open question. M cells or others. Yeah. Yeah. So we have to be cautious, though, about bulk interpretation for rare or non-rare cells, because they can miss a lot of these compositional differences.
I think it would be great to just follow up on dynamic versus sort of the proportion of cells. I think that's an important point, and maybe just taking a step back in terms of calling things rare. They might be rare because we never found them before, and then once we find them, we know how to look for them. So that, and of course there's the number of cells and how they're expressing or what they're expressing, and the levels of expression of some of the genes that might be signature genes for those specific and rare cells. So I think just to take it back to the first question, one thing that we always ask from a perspective of someone who's focusing on one specific disease is what is the real function of the gut, and what functions are eventually disrupted or changing during disease, and what are these functions in health. And if we want to understand sort of the specific areas of the gut, and for us at Hamsley we're focusing on ascending colon and terminal ileum, but many of you expressed interest in other areas. So are you thinking as sort of the function of the gut? Are you taking that into consideration? Is it something that you think about when you're analyzing your data? So what is separating sort of your single cell and what that cell is doing from the function of the entire organ or the tissue? So maybe that's a question we need to start tackling if we want to define questions and goals and outcomes. So any thoughts? I know we're shifting from the overall
no, that's fine.
list of questions, but anyone? I mean, I can give you an example. Absorption. So the gut absorbs. Do we think about it when we're analyzing our data, comparing Crohn's and healthy individuals? I don't know if that's the case. If we're looking at a single cell analysis data, I don't know if we're taking into sort of, we have it in the back of our minds. We studied it in medical school or elsewhere, but are we looking into what are the functions of these specific cells in absorption? So maybe it's not sort of a disease itself that is starting, maybe it's sort of a dysfunction in one of those sort of characteristic areas. I see someone who just jumping in.
John. No, absolutely. I mean, that was my main interest in kind of when we first started working on some of the projects in IBD and namely ulcerative colitis was, can we actually define what is wrong with the main principal function of these epithelial cell type subsets? And then I guess your question of function is more getting at what we would define the state of that cell and what it's doing. And I think one of the challenges there is that while we think about gut function in terms of physiology, we maybe don't have the molecular parts list for what some of these core functions are that has really been well defined and discretized. Or at least I have yet to find signatures that are definitive in terms of functional roles. And this is something that probably requires expert opinion and thoughts, and then of course experiments to validate what some of these gene modules or metacells might be actually doing. I don't know if others have thoughts on that as well.
But I wasn't sure what in the context of the creating the Gut Atlas 1.0, whether the first step is really to get the clearest possible view of the cell types that are involved and their transcriptional apparatus, and then that issue about how that contributes to the physiology or the function is maybe a second order question. You probably need all the bits together first because it's before you can answer that second one. But maybe that's a bit unambitious, but I figured that probably, at least from the bits that we're doing, we need to get the cast list in order before we get the kind of script really tied down.
But how, so when do you stop? This is a question that came up earlier. So when do you decide that now it's enough of creating that list and we can start thinking about what's wrong in the state of disease? Because if I understand correctly, and I have both Sarah and Aviv looking at me, so if I understand correctly, the purpose of the Human Cell Atlas having sort of a reference map for the healthy atlas is really to address the needs that we have in diseases. Because otherwise, why would we study sort of just the healthy?
It's for the people at home too.
Yeah, exactly. The people at home. Thanks, Jose. So I think even if you withhold disease for 1 second, and I have the greatest interest in it too, if you want to understand homeostasis, or rather, if you want to understand disease is in many ways breaking down of homeostasis, and so the normal tissue on its own, even without being a reference, could be a great way of understanding homeostasis, especially a tissue with so much external insult all the time. So it definitely has to kind of bring itself back. So I think the question Garabet asked is very profound, because there's a difference between saying, I want a catalog of all the cells that are there, and between saying, I want to understand how this tissue works. Because for the tissue to work, the different cells work together. And that has a deep impact on how we collect data, because if we're cataloging cells, we do it one way. If we want to understand how the tissue works together, for example, then spatial measurements are going to matter a lot. And also knowing, where you are, right, along the anatomical locations, because these anatomies are actually performing different functions. And please don't ask me which part does what. That's kind of the end of my knowledge. But they do different things, right? And it has impacts. You look at the enteric neurons, which I think are verifiably a rare cell type, especially the subsets, then they're different in different locations, because those different locations are performing different functionalities. And so having that connection that puts the cells together, I think, is important beyond the parts list. Also recognizing that by now, 200 and something individuals and 1 million and something cells have been collectively profiled. Yes, not all of this is published, but let's assume it will get published in the near future across these many different labs. We can assume that we got a first pass census. It's not full, and there will be areas that there would be special things, and there are gradients, and most of them are mucosal, so they didn't actually transverse. I think there's a lot less there that goes in, but it's a lot. So we can start thinking more ambitiously about beyond the parts, because anything we discuss here today, by the time it kind of gets granular enough that we're doing something, it's down the road from now.
So we have a home user. I will be pronouncing all the names wrong, but Sandeep Kumar. She, I think it's a she, or no, it's a male. Okay, so he's working as a postdoc fellow in LTRI in Toronto, and he's developing new statistical and computational tools for inferring developmental programs for single-cell data. And he says about the topic that you are discussing now, that inferring dynamics from single-cell data can also help us understand better the mechanisms regulating gut plasticity in homeostatic conditions, as well as after damage.
I just wanted to add on the point that she made, Aviv, and especially you, Paul, as well, saying that maybe the version 1 of the aim is not that ambitious, but I think it's a really important aim as a pathologist to really have a good knowledge and a catalogue, however unambitious that may seem, of what there is, and also in what amounts there is across the gastrointestinal tract. And the reason why I'm saying is that, for example, if you take Paneth cells that you're allowed to see them in the small intestine and in the right side of the large intestine, but if you're starting to see them on the left side, that's abnormal. And so really being able to characterize what there is in normality, and also quantify that and in normality, and perhaps also across ages, because we know that that changes as well. That's quite an important task, although it may not sound so ambitious. But I think it's important before we start saying, oh yes, there's increase in the number of Paneth cells on the left-hand side, and that's associated with whatever disease, etc., etc. So that's actually, although simple, but very important task to establish that framework of normality.
Yeah, so I just want to kind of take one step back and also emphasize that the Human Cell Atlas vision is to map the healthy normal reference, and that in itself, to kind of emphasize that in itself is actually an intellectual endeavor, curiosity-driven science that's worth doing, and that, as Louisa emphasized, ultimately will have implications for understanding disease. And so yes, there's the motivation of disease, but there's also the motivation of actually understanding our bodies at molecular resolution. And so the 2 points that have been mentioned before is the spatial understanding of tissue architecture, and I just think that's where there's still a long way to go, and it would be great if people could kind of chime in with their thoughts on that and what's the most pragmatic route basically to achieve that. Jeff is here, it'd be good to hear your thoughts, Jeff, on that, and then perhaps also on how much donor variability is necessary. Louisa mentioned it in terms of across the lifespan, so pediatric, adult. Development is obviously, there's a separate session going on in parallel on that, but what are the thoughts on genders, genetic diversity? I mentioned the Human Genetics Project going on with hundreds of donors of terminal island rectal biopsies and so on. How important is that? So it'd be great to have some discussion around that as well, and thoughts on that front.
Jeff, do you want to chime in on the...
And I think this is actually a pretty exciting time for these spatial approaches, because there's now a suite of these approaches that have different pros and cons that can basically be some more readily adopted by others and others. So there's a little bit of development phase to get these out there. But I think we're looking at a period of time where getting these technologies out and deployed in a variety of tissues, such as the gut, is actually not very far away. And so I think now is partly the time to start thinking about what these atlases will actually give us. Can we go from spatial organization, which is obviously important, to an understanding of the functional interactions that organization shapes, which I think is the critical step to go from the catalog, from the organization to a functional tissue. And I think that latter question is an important one that we want to start thinking about now in preparation for the data that we'll be collecting, I think, pretty rapidly quite soon.
You need to push it up.
Yes, you switch it off. Okay. So I fully agree with what Aviv and Zara, and you were saying that we need to have a 1.0 health. But I think by studying health and disease at the same time, combined, is going to tell us a lot about mechanistics, about how it derails, what is changing, much more, I think, than just at the moment looking at the healthy tissue, because we will be staring at a more or less in undynamic state. We get a state out of it per cell, and that's it. And okay, we can say this cell sits next to that one, and I think that's very interesting information we're also looking at. But disease and health next to each other, I think, is really bringing a lot. And if I hear around what people are saying, that everybody is doing healthy and disease at the same time, or more or less, because it brings so much more information immediately. So I think 1.0 perfect, but we should concentrate a lot more, or at least not disclaim that it's so important and we'll do that in a couple of years from now. Once we know enough about healthy, I don't believe that. I think doing it in parallel is much more powerful than doing each individual.
I think what will happen in practice, and I think it's actually happening right now, is that the difference is that in healthy, you can think about a comprehensive kind of top to bottom type of analysis of an organ, like what Ed described is happening now in the brain, right? You just go and you get the whole brain and you get all of it done. Whereas in disease, it's going to be, it's not that it's haphazard, it's the nature of disease, that this person gets the lesion here and that person gets the lesion there, and it's a little bit like this and it's a little bit like that, and so you would get those collections as well, and they will happen together. They're happening together. Even the examples that Sarah and I showed today, the vast majority were hybrids like these, where you have a bunch of healthy and a bunch of disease. But what the healthy can give us that is very difficult for the disease to do is that it can give us this top to bottom comprehensiveness, so that now when you get disease in this locus versus disease, just not exactly in the same place, you can actually compare them. And one of the things that to my understanding, is true in the gut as well as in other conditions, is that it's not exactly the same disease when it's not in exactly the same place. And that there's often very little understanding of the basis of that. Sometimes people understand really well, and sometimes they don't know at all why the right side disease and the left side disease are so different from each other. So that's what the healthy reference, I think, gives you opportunity. And that's why one can think about the healthy reference as something like that that really takes you through, whereas in the disease, you get this kind of patchiness, which is just the inherent characteristic of disease is that it looks like that.
I guess, can I make a follow-up point about the healthy aspect? So we got quite fortunate with in the ulcerative colitis study where healthy was something that we could get from screening colonoscopies, but it was age-matched-ish relative to the people that actually have the disease. And then there's efforts to kind of span the age spectrum and to look longitudinally throughout the gut, both spatially and temporally. And so the main question is, what as a community are we willing to kind of accept as a true healthy reference standard? Because the same screening colonoscopy is not happening in a 10-year-old, although we can get early Crohn's disease from a 10-year-old because that's clinically indicated. So then where are we kind of drawing the line as far as reference healthy standard and true healthy standard?
That's a big question. It's a big question.
I wanted to say something about the previous comment, but also responding to that, I mean, I completely agree it's difficult to get the right control, but I think that we're trying, I think that people are trying more and more to do that by, there's a screening colonoscopies for other conditions, like chronic diarrhea or symptoms that are related, but they turn out not to be IBD, and I think that's better control than the older, population that is screened for cancer. But I mean, it definitely, like we're trying to do pediatrics controls, and it's really, really difficult to get those. So, but I just want to, before I pass you the microphone, which is not working, to say that regarding the issue with the healthy, even though I agree that I like to see the disease, the healthy controls, I think I agree that they're very important because of the homogeneity. That I think when we're learning about a tissue, having the control, the healthy tissue is really good to understand pathways and understand cells in a much more homogeneous environment than when you go into disease. Patients are very, very heterogeneous, and doesn't matter what you look at, even if you have something very, very simple, you see this huge heterogeneity because patients have these intrinsic different mechanisms that are very actively changing, and also in between patients. So I think that's also something that controls bring that will really help us to have like a standard to look at.
Hi, And for the reference, I think, yeah, it can be very heterogeneous from different people or from different sections. And also, people already noticed that the cell proportion change may be the major feature or the consequence of the disease. Actually, I just want to notice that the cell proportion change could be depends on how you process your data. Because if you select the cells, maybe such as you use a threshold of 500 genes or 1,000 genes, you will get quite different cell proportions.
Sorry, did you mean, just to clarify, that how you analyze your data will define eventually how homogeneous or heterogeneous samples are from each other, or individuals are from each other?
Even for the cells from the same individual, because some cells, such as the T cells, you will typically get less UMIs than epithelial cells. But if you use different threshold, you will get quite different cell proportions.
So,
I think the point that Yeru is making can also be thought of. You can say it's a proportional difference. If you took two things that vary in their expression and you said, here's a category and here's a category and I'm seeing more than this and that. Or you could say, it's one population and it's shifting its programs. And now all of a sudden it's an intrinsic gene expression change and it's no longer a proportional change. That's driven by how you decided to analyze and interpret your data. It's the exact same data underlying it. And we do see that. We see that there are, and a lot of it is kind of field dependent. There are fields that believe in very strict categories, and they first categorize things, and then it shifts in the proportions of those things and changes in their expression patterns. And there are fields that look at things a lot more continuously, and they would talk more about how things are just shifting their expression, and they wouldn't be talking as much about proportions because you can't define. And so as Yeru said, if you move the threshold of when you categorize two things, you've changed the answer. But biologically, nothing actually changed. It's the exact same data.
So going back to our strategy, so what I'm thinking is that indeed, as Aviv already mentioned and Jose also mentioned here, is that it's very important for us to understand the different location in healthy individual first, and then go back to the disease and look at focal sites in which the disease is manifested. However, how do we decide which one is healthy? So we should say something like that. We should say that this is a healthy individual with no previous problems in gut, doesn't have tummy aches or something like that. When it comes to the hospital, when we take these samples, this is a healthy individual for us. Even if it's 70 years old or even a 10 years kid. This is one. And the second thing that I was wanting to talk about is that maybe the differences in the location between the different cells is coming from maybe not really human factors, but maybe also the environment. For example, we have to also pay attention to the diets and maybe the microbiome. So maybe, and this is a question that I'm facing here to everyone, how can we integrate these data sets, now the diet and microbiome, with our single cell data from healthy individuals? And how can we do it simultaneously together from the same individuals? If anybody has something to say about it, I would be happy to hear.
I'll just jump in to go back to something that Paul said earlier. Maybe we need a first draft, or we need to start somewhere. And if you start layering these on the top, it might complicate your interpretation, or at least understanding whether a healthy, because the environment changes very fast, and especially if you're thinking about equity and diversity in the samples that you're using to build an atlas, you might not be able to actually factor in all those environmental and microbiome factors. However, it does not mean that it's not important when you start shifting to study diseases or look into a special phenotype and location and so forth and so on. So I think it's a second step. It doesn't mean that it shouldn't be done at the same time. And I add on top of that the genetics, because I think it's an important component that really affects how these cells actually, which state they are at, how they interact with each other, and where they are in the spectrum of the tissue, entire tissue, or the gut itself. So it's a big topic to discuss. However, I don't know if it is within the setting, or maybe we should add, moving forward, the microbiome and the environment as components. Hopefully, we'll get some idea on the environmental sort of factors when we start looking into a single cell ATAC or other technologies. Jose, do you have?
Yeah, so I guess in kind of jumping off of the last few points that were made in Aviv's, as far as some fields where things are defined a little bit more discreetly, and I haven't heard screams from the immune cell room, so I don't think they're talking about macrophages and dendritic cells yet. But, you know, oftentimes when we're looking at the intestine, in single cell space, the immune cells, to a certain resolution, jump out quite cleanly. The stromal cells even jump out quite cleanly. But then when we're looking at that even healthy epithelial smear, the question becomes, where do we start to draw lines, and what do we call each of those? And each of us have a different kind of operational definition for that particular manuscript as to the level of resolution that we kind of defined to. And so I don't know if there's any efforts amongst the field here to kind of actually sit down around a table, as we are today, and start to discuss what is that common core language that we want to develop as to how we can define even those healthy cells and their roles, or what the best way is that we can kind of get best practices out for investigators that are interested in using reference data sets and kind of comparing their own data to what HCA efforts are also generating.
That seems like a really important kind of outcome from this, because I think just looking at it from my own very limited perspective on the immune side, we're quite familiar with trying to designate cells according to a bunch of phenotypic things, which have got more elaborate over time, but that's effectively what people do, and assign functions to them, which I guess is kind of speaking to that point. But maybe so it seems really intuitive to me that's the way to go. But probably for all the other cell types, it's much less of a historical path. So maybe if we could just bring everything up more or less to the same standard, perhaps not as kind of obsessively splitting as the immunologist, but up to a kind of relatively good.
Gets there no matter where you start.
But yeah, so I think that would be good, and maybe we could use this sort of as a way of getting everybody to do that.
I just would like to comment on how to define a healthy gut. So I just would like to say what we are doing or planning to do. So first thing, endoscopy for sure, and also looking at calprotectin levels, CRP levels. These are the things that we are really aiming at looking. But I don't know if anyone has other comments or perhaps serological tests that should be taken into consideration as well to look at general health status of these subjects.
Maybe two points. One, back to the healthy question. You don't do colonoscopy on a healthy patient. There has to be a reason. So healthy, what is healthy? Right? That's one. The other is the microbiome, definitely interesting and definitely important. But from the celiac, and I think Paul may have the same experience, the cure, so to say, is gluten-free. If you look a year later, only very few patients have a normal intestine or close to normal. 8 years later, 50%. So measuring the microbiome and its influence may not be so easy because you can't change it quickly enough, and the reaction in the tissue is not strong enough or not immediate enough for us to really determine it, I would say. But that's out of celiac. I don't know what in other cases happens.
talking to the colonoscopist and gastrointestinologist, I understood that when there is a remission for IBD samples or IBD colons, what happens is that most of the time the remission happens around the same region of the gut. So that means there is some memory of the previous inflammation in the gut. So that might be also something to consider if we can find out within these samples how the memory or epigenetic memory can bring back the remission back to the same place.
Well, I think it's another argument. I think everybody's agreed that we've got to get it seems, the basic normal healthy data as a decent resolution, which will give you some part answer to that disease. I mean, obviously it differs from individual to individual, but it'll give us some idea about the regionality at a sort of macro level. And then we could layer onto that the disease, hopefully with some extra knowledge. Because I think Aviv is right, the disease, from the immunological point of view, it's really important because we can see all the facets of the immune system laid out before you, which you can't always see in the steady state, but it's going to be really patchy. So because it's opportunistic depending on the disease. So if we take that view, as you say, of really getting the mapping in a healthy state, we might be in a really good position at the end of this, at least first phase.
So maybe we can use this to transition and look back actually to the list of questions that we had, just to try to cover and address some of these. And we heard a lot this morning about the gut and eventually how many samples to collect. We know that there's a lot of and many projects now ongoing, and many others that will be soon sort of announced that are also ongoing. I think it will be great to maybe tap into sort of standard operating procedures, because we heard that, for example, sampling two biopsies very close by in the same location, same tissue, or same area in the gut would be beneficial moving forward. So what are your experiences? Do you have any ideas on how best we could sort of standardize protocols and share them? The freezing example, sort of freezing samples and processing them later was a good start for that discussion. Can we sort of expand a little bit more and get more details to help everyone and report back this afternoon? Go for it.
Yeah.
Freezing is a process standardization, which is definitively very helpful. It also helps for, sectioning things later. There's a lot of stuff you can do more easily in a frozen setting than with a wobbly tissue. I think the other thing that is challenging, and I even alluded to when I talked, was the fact that unlike some other tissues in the gut, there's vast territory where it's hard to say that you took things from the same place. And one possibility might be that when we actually get things like single-cell profiles out, then those would help you understand where you are relative to each other. But I think it's an open question on knowing that you can say, I took it between this and this, but you can't just measure in, say, centimeters, because the scale is different in different people. And it's not clear that there's a lot of landmarks in between, at least from our conversations with folks like Garabet and others. So to me, that's going to be one of the challenges, because if there is a gradient there, and you're not sampling from the same position in the gradient, and you don't understand the length scale of the gradient, and some of this gradient is more composition. Most of it is something that's smooth, but there's some other populations that are more discretely changing. Then you take from one person from another person, how do you know you took from the same place? I think that's, and then you would say, oh, people are very, very variable, and turns out they're not really very variable at all.
So would some of these challenges be addressed by sampling postmortem?
So possibly, if you sampled some individuals postmortem, that you could take more like this, right? Like with some intervals, and you could say, okay, that's what it looks like. Now, as I have a more sporadic sampling, I can peg it to something. And that's really what people have done in the brain. They've built themselves, it's people like Ed Lin, they built these amazing reference brains, well before there was single-cell RNA-seq, right? And now they have some sense of where they are. It's even there, it's still difficult, but at least they have something.
So do you think of if that you could get a sort of essentially a gradient score that you might be able to assign to a given bus? Well, you can do it within a crypt, I think, already, but that's just... Yeah. But you have the longitudinal gradient.
The question is there a gradient approximate to this?
But that should be testable.
Right, so in Moshe's old paper with Adam in the small intestine, there was a gradient. It was really and you could see it by just sampling, in 3 regions, and whatever you position there were shared features. A stem cell is a stem cell, different, but the stem cells, proximally and the stem cells distally are not actually the same. They have some other features that distinguish. So that's a mouse, not a human, but it still, to me, it was very instructive, and whatever surgeon in the brain, I just want to point out this, after brain I should do it this way.
Is that when they first started seeing things that now we understand are on a gradient, those cells in gene expression, because they were very far, were considered to just be a lot of different subsets. People didn't realize it was a gradient. It took a while to understand that. And it was only as the gaps were starting to be filled that it turned out, oh, they're ordered like this. And that goes with the layers and so on. Yeah, in some places, like in the dentate gyrus, people are still arguing whether it is a gradient or it's not a gradient. So, but definitely at first, if you're far enough on the gradient and you're taking from 2 different places, then in the brain, the answer was it looked like different things. They didn't actually understand that they were connected on a gradient.
Considering the building of the version 1.0 of the Human Cell Atlas, especially if we consider the samples that are coming from postmortem subjects, how worried should we be about the fact that different cell types may die at different rates, and how this could bias the final results after all? This is a concern that I have, for instance.
Oh, that's far. Okay. Go quick.
Within the tissue. That's the best we can do for now. I said so this is why we are trying to combine both postmortem resections and biopsies in order to really try to understand whether we can really recapitulate everything in postmortem. If we can, that will be the best solution. If we cannot, then we'll have to use also resections and biopsies in order to fill up the gaps. That's the best we have.
And how to set if you can compare the samples during the quality control of the sequencing itself? Where to put the cutoff? These samples can be compared or not?
Yeah, so we already, so as Aviv already mentioned, we need the bulk samples as well. Mm-hmm. So this is give us a guideline whether we are somewhere almost there or not. And of course, we will never know the real answer. There are probably some rare cell types that are still evade us, or cells that really die really fast. But at least we'll have a nice picture of what's going on there.
While it's been passed, I should, from the immune side, there's a whole bunch of resident memory cells that we just didn't know about when we did grinding up stuff, essentially, for years and years. And basically, well, single cells, it's really obvious because of just the power of it. But the spatial approach using in situ is also really going to help address that question too. So, I mean, if any other cell is like those resident cells, then it would be the way to go.
So I just want to add a couple more points about what you said about postmortem collection. So taking samples from, obviously, during colonoscopy from patients, usually people don't have colonoscopy unless they have something wrong with them. And so they're not exactly normal. Postmortem collections, again, so coming back to your point that you made about when is, how much variability is going to be and the timing, etc. And actually, it's also why the patient dies. So if somebody has like status epilepticus or they have sepsis, all of those factors will have an effect on the state of the cells in the gut, across the body, but particularly in the gut as well. And the other point is even if you take not postmortem but transplant donors, because there is an option for that as well. So again, usually transplant donors will be on supporting machines, and that in itself will have an effect. That stress will have an effect on the gut, across the gut as well. So that's something else to bear in mind. But the other point I wanted to make is that should we consider about establishing or generating a catalogue of a spectrum of what's normal? So, for example, what I'm trying to say is, if we take blood sample from a patient and look at hemoglobin levels or glucose levels, whatever, we don't go just for one number. It's 5.5 and that's it. If you're below or above that, that's wrong. But really, there is a spectrum that takes into consideration different racial features, ages, weights, etc., etc. So perhaps I'm just suggesting maybe we could generate a spectrum of normality, if that makes sense.
Sounds very sensible. I guess it's just a matter of scale, isn't it?
Of course.
Because those blood samples are done on thousands of people.
Because all of these factors, during postmortem collection, transplant donors, colonoscopy, there are all these factors, but there will be somewhere in the middle. There will be the spectrum of normal, and I think generating that might be useful as a basis. So.
Well, hopefully, I don't know what. Yeah, go on, Niels.
Yeah, well, I guess to get back to the question where we started up, which is what is the question that we're looking to answer, and I think that what we're probably coming to is that each sample or each type of sample that we can collect from or procedure we can collect from is geared towards answering a different question, and that only by combining different types of sampling modalities can we really get at both the longitudinal sampling, but also the healthier sampling, maybe from a few select sites, and then start to understand where along that gradient, both of relative health and also position in the intestine, are our individual cells lying. And so it might be that we need the postmortem samples to have that kind of gradient construction, and then we can take the few incidental true healthies we can find either from screening colonoscopies or consented biopsies in certain age ranges, where we can start to build that out. And there may not be as many of those. They're obviously very precious samples, and one needs to kind of think carefully and also ethically about how to use them. But I think that not one data collection modality is going to answer all of the questions that we have.
So having a sort of a checklist of what is acceptable to be called normal, I think we're doing it in many efforts in the HCA, at least things I've been involved in or meetings, even for the ethics. I mean, you have a checklist of things that you could consider to be acceptable, and probably for normal or healthy tissue, we need to develop something like that for the gut and consider that would be the range of the spectrum. Jeff?
I just have some thoughts about how to tell the disease-associated genes is related to the region difference, or really related to the disease induced difference. So, for example, I'm thinking about whether we can use the healthy samples from different regions to construct a to order the cells in pseudo-temporal orders. So instead of putting them into a discrete, we order them in a continuum. And then we identify the genes showing the changes across these pseudo-spatial orders. Then then these genes can kind of a landmarks to tell whether this is in the proximal region or whether it is in more distal regions. And then using this gene set, we can infer the possible regions of from the disease samples and the patient sample so that we can somehow align them into the same spatial. And after this spatial alignment, we do the further gene expression comparison so that we can know whether this differential expression is still, we can somehow, then after this alignment, then the DE gene can be more likely to due to this disease rather than this spatial difference. I'm just thinking whether it is possible to do it this way.
I'll defer the question to a spatial specialist rather than I won't be able to answer.
Okay, so I can try and field that question, and then, but I mean, I think that type of referencing is probably hard to infer how easy it would be beforehand, because it depends on the scale over which these gradients occur and their strength. And I think sampling across multiple sites and keeping track of where those are is going to be the way to get at that first order answer to that question. And so that actually segues nicely into what I was thinking. So the question I had, and this is a very naive question for the panel. I come to this organ in a very different way than many of you. And so it sounds like there are great questions associated with we need to have a broad range of samples from geographic sites, from healthy state, from ages. And also we'd love to be able to pass these through multiple modalities. So the question is, when there is an opportunity to take a sample, how easy is it to collect multiple tissue and store that tissue? I mean, there was a little bit of conversation at the beginning about how well things can be frozen and then brought back for later analysis. But certainly if it's possible to get excess tissue, I think it's not unreasonable to now start banking tissues for spatial methods, because these will be well available, I think, in the next year or less. And it allows you to then use the same samples to come back and add on modalities. The contrast to that is, of course, if it's easy to get these samples, if you're always seeing patients, then perhaps there's not much benefit to that. So that's just a naive question for... Please.
I would say that if the tissue amount is not limiting, which is often the case when you have resections, you have a lot, you absolutely should bank. We bank all the time. The benefit of banking is, a patient actually donated that tissue. We should treat it with the utmost respect. The second insubstantial reason is, for that tissue, if you collected any other molecular information, you'd rather have extra for that, get more bang for your buck. So I think it's the question is whether freezing is something where the specific protocol for freezing and manipulation and preservation of the tissue needs to be something that we're advertising more strongly in our community so that people actually bank good tissue. Because it's often not just drop it into, there's something like you have to get rid of the ice or the water or something. So I think that's or you have to put it on a slide, or you might be able to section later if you preserved it like this. I don't know more than that, but I know that you actually need to give people instructions.
Yeah, I think that would be an ask for kind of all of the communities, and actually more for the technology working groups, because people who are really at the cutting edge of each of these fields know what works best for preservation. And when I think microscopy, I think, oh, I might just dunk this in OCT and then zap it and come back to it later. But I'm getting the thumbs up, so that might work. But for something else, that might be horribly the wrong thing to do. And so actually having a kind of best practices for pick your favorite method would be great as far as sample preservation and storage, because I think that's something that would be a big resource for any of the tissues. Not to mention tissue-specific protocols, which of course then the taxonomy ever grows. But in general, a starting point.
There's a question.
As for the reference, I think, of course, if you have a better, more comprehensive reference, that's much better. But we should notice that the RNA sequence just looks like some analog signals. So it's noisy, and also it's influenced by some other factors such as circadian or this kind of factors. So so if we have a reference and then we found something, if we map our cells to the reference, we found something that are different, I think that we need to investigate more. So, and then we can add those to the reference.
I have a question regarding the storage of samples for Spatial also. So I think the gold standard by now is embed it in OCT and freeze it right away. Has anyone tested, what about collecting these samples in a buffer with RNase inhibitors and then proceeding with the OCT embedding and freezing? Does it help? Has anyone tried?
We haven't tried that. I don't know if for spatial transcriptomics, if you guys have tried things like this. Our standard go-to is to drop a tissue block in OCT and freeze it. We've done heavily fixed tissues as well, but not carefully enough to say that there isn't some performance drop, though it can work. And then I think that the big worry is that these experiences are drawn from a limited set of tissues and limited size of the tissue components. So, I think if tissues are precious, then we have to think carefully about this. If there are instances where you get large tissue amounts, then it might be reasonable to try preparing some in different ways, different sizes, different preservatives. I think this idea of an RNase inhibitor is a very interesting one. And then we could imagine now systematically understanding the preservation of these tissues in these different methods. But I think without doing studies like that, it's hard to say for sure what would be the best approaches from at least my limited experience.
Just one more question is regarding your experience, a block frozen -8, for how long do you think the RNase is stable? Is there an answer for that?
Yeah, no. I mean, again, this is something that we only have a limited amount of time where we've been testing this. In mouse brain, I can tell you 6 months is fine. I suspect longer is definitely fine too. I've done some look through the literature at RNA integrity values. It seems like many tissues can be maintained at -80 for quite long times, but it's difficult to know if that holds true in all instances.
Just to be thoughtful on time, we have less than 15 minutes to wrap up. Just to continue on the spatial discussion, one question that we had initially to cover today, and hopefully we can just touch upon it, is to sort of think about integration of spatial data, because now we'll get more and more of these with single-cell RNA sequencing, which I think you referred to earlier, and what do we need? So AI, machine learning, and how we can do this.
I think that's 15 minutes is not enough for that topic. I think it's
2 other points to cover.
Yeah. Okay. So 3 minutes is not enough for that topic. Yeah. I would say, I think we don't know what we need. I think it depends on what we want to get out of it. Certainly, in my experiences, you can learn quite a bit by relatively simplistic comparisons, but there's a tremendous amount of promise in leveraging the correlation structure in these different data sets. And there are efforts underway to tackle that. Whether AI is necessary for that, I don't know. But I think the more we can get multimodal data sets of the same samples, the more we can really start to build and test these new analysis approaches.
So just to add on that, how would you integrate, because thinking about this morning's presentations about the roadmap and the dynamics, so how would you integrate those different versions or options that we sow into data analysis, because I think that's an important component to keep in mind.
Yeah, I think it's a very difficult question. So I think there are kind of tiers to that question. The first is that you essentially analyze these data, at least to some degree, independently of one another, and then do a comparative analysis at, say, the level of cell types, and then extract insight from that. Or I think the alternative, which is perhaps even more exciting, is to try and leverage joint analysis of these data sets. And there's been some great work that's starting on these directions. And it's hard to know until we see how those ideas develop, how much more you really gain from that approach, but I think there's a lot of promise there.
I think you've come to the right session in terms of biopsies, though, because just going back a couple of steps, the gut people will take multiple biopsies, in a biopsy setting, multiple. To do it in the liver, if you got 2, you'd be really grateful. So you get 10 from a site. So I think there's always going to be tissue that could be taken forward, and we really need the spatial transcriptomics. So I think it's an ideal combination.
Yeah, that's what I was just about to say, because I think a lot of the problems that have been raised in a way are in fact opportunities that exist in the gut and not elsewhere, where you're not incidentally taking a dozen different biopsy bites as you move your way back out. But along those lines, the question becomes one of length, where even if you take 2 adjacent biopsies, sometimes you get fortunate and it's more or less the same, or other times you're like, I hit the jackpot and it's only T cells and B cells here. Where'd the rest of my cells go? And so the question is, when you hit a sample like that, how do you address that? Is that noise, or is that something that you somehow need to, you can't just discard it. So how do you kind of address that variability and, I saved one for microscopy, I saved one for single cell RNA-seq. Now I have 2 completely discordant results. Is that technology, or is that health or disease that's kind of driving that? I don't mean to be speaking, you just have the microphone now still, but anyone else, yeah, just.
I think I only had 3 minutes was the idea.
That's why you're supposed to look at lots of different individuals, right? To try and ascertain whether that was just random chance. I think this is the structure of the gut, that you have those isolated lymphoid follicles, and you have very different structures, and that's inherently tissue acid. And you need to have, ideally, what we would like to do, right? Money was not an issue, amount of data was not an issue. We would have to have as many biopsies as possible for anything we want to do. But the reality is that even if we're lucky, because we get biopsies, still you always want to do a lot of, you want to freeze for these, and then you want to have, maybe look at the microwave, and you end up with 2 or 3 biopsies to do for one analysis. And that is a very important issue, that they're very different. And if you go to the disease issues, look at the pathologies results from Crohn's,
and especially in Crohn's disease, is super highly patchy, not just in patchy lesions, but even histologically. So that issue is a big issue. And the only way, in my opinion, to reduce that is having, pulling more samples together, because that variability, you cannot say this is a wrong biopsy because it has a lot of lymphoid cells, but that's a very important structure in the gut. You want to see that too. So I think that's intrinsic to our tissue, and ideally we will want to have more biopsies so they represent more what we have. That's ideally. I know then the limitation of how much you can use for everything, it's big.
So just to go back to actually Orit's presentation from this morning or this afternoon, so having 2 biopsies and then pulling them will give you a better sort of depth. Sorry?
You should tag them before you pull them.
Yes.
So you know
Yes. Absolutely. That's more the technical aspect that I'm not that familiar with. But and that will help you actually understand better the depth of the samples. And the third phase, which was the breadth, this is where you go and replicate it in multiple individuals and just to verify and validate whether these are sort of the location and the state and the cells the types of cells. So just to go to the last 2 points, which are sort of related. One was the topic was on pathology and the diseases, and we actually throughout touched upon many points raised here. And the last one was around extraintestinal studies. So what other organs or tissues we should be studying in addition to the gut? I mean, we have some scientists here or clinicians who are working in the liver or pancreas or other organs. So these are connected. So the gut, again, it's the second brain. It's connected to the brain and many other organs in the body. So maybe there's benefit to also think how these are affecting the physiology, the function, and to the level of the single cell. So any thoughts on how to handle disease? For us, at least from Crohn's disease perspective, stratifying patients because the disease is very diverse or very sort of heterogeneous, having a clear set of information related to the patient and the phenotype of the disease is extremely important. So is that something that in other diseases is similar? I'm sure that in celiac you phenotype patients also, or
yeah, it's a little bit easier there. I think it's less heterogeneous than Crohn's, but even with something like PSC, which is, you know, a relatively clear syndrome, then multiple subsets. So it's a bit of an endless discussion. But I think for the big one like Crohn's and UC, that there needs to be some minimal metadata that comes with the patient. Otherwise, we're going to be in a big mess, I think. I don't know if people have already addressed that.
Everyone's calm. Probably addressed it. So in terms of disease, one thing we've been thinking a lot about is longitudinal follow-up of individuals, and I think in healthy individuals is the same. It will be important to see whether a longitudinal follow-up, if you can get biopsies from the same individual, would be a great tool. I think you mentioned earlier that that's possible, maybe for cancer screening every 5 years or every 3 years. Yeah, that's. And for patients, this is something that I think it's important just to address the question whether there's sort of memory, if there's any sort of imprint of the inflammation that stays in place, and this is why maybe the disease comes back in the same area where it was in remission, and suddenly months later or years later you will get the flare in the same area. This is very common in Crohn's. It's common also in ulcerative colitis, and unfortunately, many patients lose response to their treatments and they flare up. So is this something that in other diseases is similar? Do you observe the same thing? I think everyone is.
Yeah, they might. But also the longitudinal, also because of the with the 5' chemistry, you can track the TCRs. I think that gives an enormous amount of power to track individual clonotypes. So I hope people are going to be able to take advantage of that. Adds a lot of depth to the studies, because then you can map the clonotype onto the transcriptome and get a much more. I mean, I'm feeling bad about not being able to address your question about function, because that's ultimately the kind of critical one. But I'm not sure if we've got a conclusion yet about how to tie it all back together to create essentially a functional atlas. That seems like a really amazing aim.
I think that's a big aim. It's going to take some time for us to figure it out, and probably the first data that now are being published and generated will help us start crafting a roadmap for including the function or the functions of the gut. So, any last-minute questions from you, from everyone? Anything that we haven't addressed and touched upon?
So listening at all these conversations, I understood that everybody is doing their own analysis and doing different type of disease and healthy, etc. And at some point, all this will come together, and probably you will do some integrative analysis where you want to see and compare different data that is generated by different part of the consortia. But then it comes to the question that if we do have a consensus metadata generation, that we do know from which part of the organ we are generating the biopsies, because to my experience, some surgeons send the biopsies distal, proximal, some of them call it transverse, descending, ascending, some characterization is like centimeter of the colonoscopy, and then at the end of the day you don't know exactly where these data are coming from, the biopsies are coming from, and once you want to do the integrative analysis, then you may have some difficulties. So if there is a metadata collection form by the whole consortium, I'm not aware of this.
For the whole human cell atlas, I don't know. However, for the gut specifically, at least for the ascending colon and terminal ileum, so the gut cell atlas first launch, which is the grants that Hansley will be funding and announcing very soon. So we will have a standardized approach to actually collecting metadata. I don't know if we'll be creating a form that will be applicable to all the sites that we will be covering. Because they do have their local laws, which are very different. However, we're going to get at least annotations, nomenclature, standardized sort of language in terms of locations, in terms of phenotypes, treatments, or any other metadata that is needed. And these have been in development for many years. We've invested specifically in standard development for Crohn's disease, which will apply to many areas of the gut. And those include not only sort of basic metadata standardization, but also some omics data related language. So that is something that actually we take really very seriously, because it's very important to compare apples to apples, and I know that just looking into some of the data that exists on the DCP, there's a difference between calling it large intestine versus colon, because it might not be the same area. If it's a little bit higher than the large intestine, it might be called cecum or terminal ileum. So this is why it's very important to take that into consideration. So we have some efforts ongoing, but we're not there yet. So.
Okay, so where can we find this kind of metadata format that we can use all together?
In the future.
They're also always open to engage with them. You can engage with them very effectively to introduce tools around specifics. It's an extremely important question, because historically in genomics that has been a major failure. And I would add to that, related to what Jeff said about the freezing of the tissue so you bank it, the more metadata you collect, right, the more information you're able to say about what you did when you did the experiment, the better off everyone is, instead of you do it later on. And that includes this information like the photo, the manipulation of the tissue and so on, because maybe today we don't exactly know how to record it, but in the future.
Right. Because for us, I think it's very important as we are going to generate these 100 colon organoids and compare them with original tissue, and then you can link it back to all different type of analysis.
So we're almost out of time, and we just realized that we had a last question that was on the agenda, which relates to common coordinate frameworks, which is an important question that will probably tie it back to identifying a location or saying that this is where the biopsy comes from. We're hoping that with many efforts that are being done now with the HubMap project, as well as the initiatives on the lung, brain, and others, we'll learn lessons from these initial initiatives, as well as actually trying to learn lessons from what has been done in mouse models for the gut specifically, and eventually translate these into a human, and hopefully those will be approaches that we could use moving forward. I know we're short on time. Any other last?
No, I think we probably, well, I'm not sure we answered any of the questions, but we did talk about them all. And we'll try and pull that all together. I think there were some common themes that emerged. We didn't quite get onto the host genetics aspect, but maybe that will come up in some other talks. So,
yes.
Yeah.
There's a lot to cover in a very short time. And I think what's clear is that we're making lots of progress. And I do see it just being involved in the Human Cell Atlas initiative for the past 2 years. I do see already a huge, tremendous progress. So with that, I think.
Yeah, we can stop. And yeah, thanks.
Thank you.